Hello and welcome to everybody. I'm your chair, but before I ask the speakers, I'm going to say one thing. Today is all about success, how to achieve success. And so I'm going to say the one word about failure. I got my first grant in 1981. How long ago is that? Almost 50 years ago, and have had a multiplicity of successes since and millions of euros. But for every single success, I have had at least 3 failures, at least. The most recent one being last night. So, to everybody, this is about keeping going. Going again, going better. Fail, fail again, fail better, as Beckett said. And this is why the lesson from Guinness's is just so wonderful. It is by keeping at it that we achieve excellence and high quality. So, to go back to the success, let's hear from Rebecca Corrigan, who got the Lister in 2018 and is now in UCD, and presumably the Lister was really important in that job. She's going to tell us.
Can't really see that, but yeah. All right, so we're here today to promote the Lister. And what I wanted to do was to use my personal story to try and convince some of you young people in the audience to apply for a Lister and to show you what kind of impact it can have on your career. So this is a career, if you follow my arrow, that started as an undergrad in Trinity, went through various different universities, both in the UK and America, for I'm now based in UCD. So going through my talk, I'm just kind of going to follow this timeline. And the one key thing that I want to point out is right in the middle there, the Lister Institute Fellowship and the impact that that had on my career progression. So my career starts in the Moyne Institute in Trinity College. That's kind of apt because the Moyne Institute was named after the first Baron Moyne, and the money was given by the Guinness family to honour Walter Edward Guinness. So there's a lot of links here between the Guinness family and great science. So I began my BA. It was a natural sciences degree, and then after 2 years you're allowed to specialise. So I specialised in microbiology. And at the time, I was very thankful to have come in contact with a mentor, Tim Foster, who was very supportive. And what Tim did was he secured me a placement during my 3rd and 4th years over the summer in the lab of Richard Novick, shown here on the right, in NYU in New York. And while I was working with Richard, we were working on staphylococci, which is a bacterium. He placed me on 2 different research papers for the work that I did that year, and kind of sowed the seed that I wanted to get into research. So when I came back to Trinity, I decided to do a PhD. So I joined Tim Foster's lab, and we started working more on this bug, Staphylococcus aureus. This is sometimes referred to as MRSA in the newspapers. It's a hospital superbug. It can kill a lot of people, but actually it's a commensal organism, so it can live in the noses of about 30% of people. And the way that it does this is it uses these surface proteins. Sorry, the pointer's not great, but there are a whole load of surface proteins on the surface of the bacterium. These are like hooks, and these hooks grab onto receptors in the nose, and that allows them to colonize and to live in the nose. So while working with Tim, it was a successful enough PhD, got a number of research papers out of it, and from that I decided I wanted to carry on in research and go on to the next step and do a postdoc. So at the time, there was a new PI, Angelica Grundling, that's her picture there on the right. She had just started her lab at Imperial College in London. And what Tim, I'm just going to see if this works. Yes, great. Right. So what Tim did was he had heard of this researcher and he invited her over to Trinity to give a talk, and basically took both of us out to dinner and kind of shoved me at her, which was a successful approach. So for any PIs and your postdocs are looking for jobs, this is something you can do. So I could carry on working on MRSA, so I moved to the UK, and I was working with Angelica for about 6.5 years. Again, this was a very successful time in my career. We published quite a number of papers together. But one of the key things that Angelica liked to do was to introduce new techniques. So she had a colleague in the University of Maryland whose name was Vince Lee. The University of Maryland, the campus is absolutely amazing. Football stadiums, car parks, just the scale of American campuses is phenomenal. But it's based at the University of Maryland, which is over here in near Washington, D.C. So Angelica heard that Vince was working with this new technique, and the technique was focused on looking at how well a ligand can interact with a protein. So basically just measuring the strength of this binding interaction. But what Vince had done was come up with a 5-minute, really quick assay to measure how strong these two things interact. So ignore how technical it looks. What he called it was a differential radial capillary action of ligand assay. But actually what he wanted to call it was Dracula, and he came up with words to fit. When he tried to publish that, the reviewers wanted him to change it. He refused, thankfully, because it's quite a catchy name. But this was a very simple and quick way that we could now measure the binding of our protein to our ligands. So having learned this new technique, we successfully published that in PNAS. But I'm focusing on the fact that it was a new technique because what I was able to do was to use it and then to shift the research question to my own area. So that's exactly what I did in 2015. I took what I had learned and I applied for a Wellcome Trust Henry Dale Fellowship. It's also jointly funded by the Royal Society, who shall not be forgotten. And I started my own research lab then in the University of Sheffield in 11 years ago, 2015. So this year was the start of my research group, but that fellowship will give you your salary for 5 years and allow you to employ a technician. I also employed some summer students, and this here is Janie Kim. She was from Princeton and spent the summer with us and then developed this lab logo, which is actually, if you can make it out, my name, but spelt using scientific equipment. So this is an agar plate, this G is a microscope, which is quite innovative. So to build your lab, there's a number of things that you need to start. And I'm sure you're all aware of this, but it is that strong publication record. Having evidence of funding also helps. So my trip to Maryland was funded by an EMBO short-term fellowship, which is EMBO is the European Molecular Biology Organization. They're great for little pots of money. But you also need that idea. And to get that idea, I firmly believe that you need to take advantage of new techniques that are coming out, so innovative techniques. You need preliminary data to enable you to write that grant, and you're not going to get that preliminary data without the support of your PI. So you need to be having conversations early on, if you're a postdoc, with your PI, and they need to allow you to work in their lab to get that data that you can take to start your own research group. And of course, this needs to be in an important research area. So very briefly, and I'm not going to go into the science, but just to say what I do. So I work with MRSA, the bacterium, and I work with bacterial stress responses. So when we're growing bacteria in the lab to study them, we culture them in test tubes, we feed them very nutrient-rich broth, so they're all very happy. But actually, that's not physiologically relevant. So in a human body, bacteria are going to encounter stresses, so the innate immune system, or pH changes in your stomach, or antibiotics. And basically, the bacteria become stressed, and they cope with this stress using stress responses. So they have ways to cope with whatever we can throw at them. Okay, so now I've started my lab in Sheffield, 2015. It's me and a research assistant. How do you build that lab? How do you take your research to that next level? So this is where the Lister Institute comes in, and very much thanks to them, they allowed me to be promoted to a senior lecturer at that time, so thank you very much. But what the Lister Institute grant, the prize, gave me was kind of immeasurable. So the next few slides, I'm just going to take you through what I did to get that grant and then the impact that it had on my research. So the funding itself is was 250 sterling. I think it's now 300, so equating to euros, it's maybe 350, which is a very decent pot of cash. But actually for me, the cash wasn't the most important thing. The most important thing was the freedom and flexibility that Lister give you in spending that cash. They kind of have an ethos where they believe in you as a researcher, and they give you the freedom to spend it in the way that will drive your research to the next level. There's also a fantastic mentorship and community feeling. So we meet in Oxford every September, and we welcome in the new fellows for that year, but also old fellows come back and we all chat about science, and it's a very collegiate and supportive environment. So there's a lot of mentors associated with Lister. And of course, every summer they provide funds so that we can have summer studentships, and those students actually get to come with us in September to Oxford and get to meet all of these amazing, incredible scientists. One other fun thing that Lister do is that when you get your award, they come to your institute and you give a seminar. But this seminar is more of a celebration of your research as well as being awarded your medal. So you're encouraged to invite your families. So here are my parents. And they came and they tell me they enjoyed the seminar, but I know they didn't understand a word. But I think they just very much enjoyed the whole ceremony of the thing. So this is Alex Markham, who was head of Cancer Research UK, who was awarding me my fellowship. So he was there together with the then director, Kate Law. So what did I do with my fellowship? I was awarded in 2018, and what I wanted to do was to use it to allow me to address bigger questions. So I told you that during my Henry Dale Fellowship, the question that I was looking at was bacterial stress responses, how do they work? That is very important. It is very important that we know how systems work. But if you want to take your research to the next level, you need to ask the bigger question. So the question I wanted to ask was, are they actually important? So I started to study whether they were important for infection and whether they were important for antibody resistance, because these are the bigger questions. So what I did was I used my Lister Fellowship to start using zebrafish embryos as an infection model for my system. So what are zebrafish? These tiny little fish here. And then this is an image of one of their embryos. And this arrow here is pointing to the heart area of the zebrafish. So what you can do is you can inject fluorescent bacteria into the heart. The bacteria will travel around the bloodstream, and you can look at how the bacteria actually start to cause infection. But of course, these zebrafish embryos are tiny, like a millimeter, so you need complicated equipment to allow you to inject into the heart. So this is what I set out to use my grant for. I hired a PhD student who started October 19. So this is Naz over here, and then these are some of her images. So this is a microscope. On the microscope is a petri dish, and in the petri dish you can see a zoomed in image of all of the tiny zebrafish embryos. Now just think about how hard it is to get a needle to inject into one of those tiny little embryos. So there's a lot of skill required here. So what Naz did was she spent the first 6 months of her PhD learning how to inject the embryos and also getting training in using human macrophages. But if anyone's kind of following the timeline, she started October 2019, and she comes into me in the first week of March, and she has just successfully completed her training with the zebrafish, and she's excited to go on now and to start using mutant strains and to start doing big experiments. March 2020. I'm sure everyone enjoyed that time. So how do you do research in a global pandemic? I was very fortunate that this occurred while I had my Lister Prize. So this is a picture of me, July 2020. The lab had been closed for 3 months, and I was allowed to now cycle back to work. So I'm there cycling with my mask. And then I'm in the lab space on my own, reformatting all the space so that we can all spread out and everybody can work in isolation. Okay, the lab is open, but the access to the aquarium is denied. We are not allowed into that space, and we're also not allowed to go over to the med school, which is where we were working with our macrophages at the time. So then now all of a sudden I have a PhD student who has gone from being happy to incredibly stressed, just like her project. And this is where Lister came to my rescue. So I mentioned to you before about the flexibility in terms of the funding. So because they trust you to do the best with your research, I was able to use the funds to set up tissue culture in my lab space. So people were able to work with new equipment. So we got a hood and we got in some new equipment. And all of a sudden, the PhD is back on track. So Naz then worked with the tissue culture and the macrophages for the next year or so. And then when the aquarium was open, she was able to go back in with a more refined question, actually, and work with the fish. And she graduated for a PhD, and she has a number of publications to her name. But that flexible funding wasn't just used for Naz. That's just one example of what I used the money for. I also used it to extend postdoc contracts. So that was Sophie who got to stay on and finish some work. I was able to hire Lucy for a few years, and she brought some really good skills in working with human cells to the lab as well, which just kind of broadened the remit of what, as a microbiologist, we were able to do. So importantly, the Lister funds has contributed to 12 publications since I started. But the impact doesn't stop there. So I've used the Lister, our findings from the Lister work, as preliminary data for 2 further awards for Henry Dale extension of my original Henry Dale, and then for a further Wellcome Discovery Award, where we're moving into looking at AMR. Sorry, antimicrobial resistance. So that is very much nearly my journey. I also just moved my lab back to UCD in Dublin. So I was a senior lecturer in Sheffield, and actually I still have 2 people in the lab in Sheffield, but I also have joined UCD as an Ad Astra Fellow a year and a half ago. So we're starting our research here. And then this is my 2 people from my Sheffield lab meeting the new people from UCD at a conference earlier this year. So what I want to do is just to stress that the Lister Institute prize isn't just a pot of money. It actually can be used to develop your career and get you out of many different sticky holes so that you can build your research. But also, this was not solely just done by me. Of course, I need to thank everybody in the lab who has contributed the data and all the different funding bodies, Wellcome and Lister in particular. So if anyone has any questions, I'd be delighted to answer. They have. They're not directly linked, but they allowed us to set up assays, and it was the macrophage work in particular that allowed us to get a lot of preliminary data, and then that has fed in. It also introduced me to a lot of different people around my department, and just had more conversations with people. And then the discovery was actually a collaboration between 6 different PIs. So it was kind of formative in just making introductions that way. And really, it's about getting your name out there and getting known as an international researcher that you can then form those links. So definitely it was vital. I think the best thing that you can do is to not be shy about contacting people and talking about your idea before you start to write it out. One of the first things I did was, there was another Lister fellow in Sheffield, so I went and spoke to him, and he really helped me visualize what it is that Lister are after, which you'll hear about later today, and how to pitch my idea in the right way. So really don't be shy or reticent to just get in touch with somebody. If they respond, fantastic. If they don't, Okay,
thank you.
Just before we let you go, Rebecca, can I ask another question? Did you find that having the Lister gave you a kind of a scientific courage to be braver with your science?
Did it reduce the imposter syndrome? Maybe it helped me learn to bury it a little more.
Yeah, but I thought the whole description with Dracula was just really great, because it sounds like, if you hadn't got the Lister, that mightn't have taken off to such an extent. And so, it's a great story. Okay, thank you. Yes.
Right.